Good afternoon, everyone. Thank you very much for being here. It's my pleasure to welcome you to the season number 3, session 5 of the Mixed-Gen series of CCAM. Today, the topic is very exciting and is related to data science in biophysics with application also to drug discovery. As usual, for the Mixed-Gen series, we have 3 speakers, 1 senior and 2 younger investigators. Zoe Kurnia from the Academy of Athens with a presentation entitled Predicting Protein Membrane Interfaces Using Molecular Simulation and Ensemble Machine Learning. And then Maurice Kahrenbrock from the University of Geneva in Switzerland, a non-equilibrium alchemical method for drug receptor absolute binding free energy calculation, the role of restraints. And then Francesco Di Palma from the Italian Institute of Technology with a presentation entitled Unraveling the Ribosome Stalling Mechanism Induced by the Human XBP1U Arresting Peptide. Without any further delay, I leave the stage to Zoe. I would like to thank the attendees. I would like to thank again very much the speakers. And please, Zoe, the stage is yours.
Thank you, Andrea. Thank you for inviting me to speak to this excellent series. I've been actually following it since its inception, and I think it's really crucial that you continue this series. It's really very important. I believe that we also have with us a number of senior scientists that you invited to this conversation. I would call it more than a webinar, because we will discuss a lot of impacts of data science and biophysics. So let me start my talk again by thanking you and by also welcome you to this new role in the directorship of CCAM, which I think is very important. And this science that I will present today was funded by the Hellenic Foundation for Research and Innovation. And I will speak, of course, about the general theme, the data science and biophysics for application drug discovery. And I will present you a case study from our lab on predicting protein membrane interfaces using molecular simulations and ensemble machine learning. So the first thing that I did, as you can imagine, before starting my talk was everything that all the students and possibly many of us are doing these days. I asked ChatGPT. So what do you think about data science and biophysics for applications to drug discovery? And of course, as a language AI model, ChatGPT responded that they don't have personal opinions, but they can provide some insights on the topic that we've mentioned. So he or she or they rather gave a definition of data science in what is biophysics, then what is data science, and then that in recent years, there have been several successful examples of the application of data science and biophysics to drug discovery. For instance, the use of molecular dynamic simulations and machine learning algorithms that has been employed to predict protein ligand binding affinity, a topic that we will also hear later on from one of the next speakers. Which is crucial for identifying potential drug candidates. And overall, the combination of data science and biophysics offers a promising approach to drug discovery and development and has the potential to significantly improve the efficiency and success rate of drug discovery pipelines. So let's see about that. So here's an outline of my talk. So I will first talk about biophysics data sources. So where can we get our data? Because data science implies that we have data. Some background for AI in biophysics. Then data science and biophysics for applications in drug discovery with a particular topic of predicting protein membrane interactions using molecular simulations and ensemble machine learning. And applications in drug discovery and some concluding remarks and thoughts to open the discussion. So there has been a data explosion in medicine and biology, biophysics lately with, of course, next generation sequencing, all the omics that have been generating, both abolomics, proteomics, lipidomics, etc. Research results are now often deposited in open access repositories, such as gene expression results, phenotypes, medical images. And we also had an explosion of structural biology data in the last decade, cryo-EM, X-rays, and NMR structures. They all came up. And of course, we had the AlphaFold contribution, which was very significant as well. Also, the global biomedical literature increases by 500,000 articles per year. And the precision medicine patient data, which in general, if we consider all of the patients are considered big data, and these are stored for each patient, for example, electronic health records. So we really have a rich source of data sets in biology and biophysics. However, these, as we will say in the end, are quite heterogeneous. So here I'm showing some data sources for biophysics. For example, from experiments, atomic force microscopy, cryo-electron microscopy, X-ray, all sorts of biophysics experiments that generate data that we can use for predictions. And that together, those experimental breakthroughs that we have experienced the last decade, as well as all the open data schemes that we are hearing, the European Open Science Cloud, fairness becoming, the data becoming fair, findable, accessible, interoperable, and reusable, and also the mandate of many of the journals that we are publishing in to publish our data in a manner that is open access and reproducible access for all scientists has really brought a tremendous increase in the available data that we have. And that also coupled with the computational power that is now available to us has brought in more applications of artificial intelligence. And yeah, so this is showing again the data explosion. I mean, here you can also see it graphically, see what happened after about 10 years ago, like around 2010, the increase has been exponential, both in the data as well in the publications. And so while these artificial intelligence systems, so we hear artificial intelligence now as a buzzword everywhere and at every aspect of our science, is not something that is new. It's not something that was discovered the last 2 years with AlphaFold. These are techniques that have been available since at least 50 years. But as I mentioned, the data was not available. Also, the computational power was not enough. So now, with the explosion of data, as well as with advances in hardware and software, we can now exploit this data using these methodologies that were quite known the last 50 years, and also the developments that are being made in artificial intelligence algorithms. So artificial intelligence systems are suitable for analyzing biophysics data because they are robust against the inherent complexity of biophysics biomedical data. They can extract trends from data. They provide models for complex processes. They can handle uncertainty. And they can also couple these efforts, as I mentioned, with literature content. That means they can combine and process heterogeneous data in a framework that can lead to better predictions. And this is a graph that I found from the internet that shows essentially how the next generation AI is powering every aspect of healthcare, not just drug discovery. And we're seeing here some of the advances that AI is helping us with. So to develop new therapeutic targets, or to discover rather, because they are already there, biomarkers, biological target identification, designing proteins, screening and generating molecules, and even up to the design of clinical trials and analyzing the data, and leading, of course, up to the final goal of the use of AI in biophysics and biomedicine, which is precision medicine, which is to deliver faster and more cost-effectively efficient drugs for patients to improve the patient lives. So the last 2 years, 2021, 2022, and 2023, has really been a revolution for AI in our field in biophysics, mainly because of AlphaFold and AlphaFold 2 from Google DeepMind, which was a breakthrough in biology and received the 2023 Breakthrough Prize in Life Sciences. We've all heard about it. We can now generate, so it can generate basically with high confidence protein folds that are not available to us through experimental techniques. However, of course, this method also has caveats, and we can discuss it in the end. For example, it cannot predict the effect of mutations, or it cannot predict structures that are not in the training set. For drug discovery, other essential areas where artificial discovery is crucial is the choice of pharmaceutical target. So which target we are going to use for a given disease, if it's a protein or RNA, discovery of ligand binding pockets, where the small molecule, the bioactive molecule is going to be designed, the actual design then of the bioactive compound. So this was also mentioned by ChatGPT, to actually predict the binding affinity of the compound onto the pharmaceutical target. Toxicology prediction, pharmacology, polypharmacology prediction, issues of side effects and selectivity. The synthetic route design, also for designing the synthetic routes of inorganic synthesis of the bioactive compounds, and of course in lead optimization, in optimizing the final lead compound for all aspects such as its binding affinity, metabolism, excretion, oral availability, and so on. So as computational scientists, we have worked, most of us so far, with traditional computational biophysics. And basically in such schemes, we have rules. For example, in molecular dynamics simulations, you have function, Newtonian mechanics, you are following a theory, you have the rules, and you have the inputs, which are the coordinates and velocities of the protein target or any molecular system that you want to simulate. And then you simulate using a computer, using these rules and inputs, and that results to an output, which is, in this case, the molecular dynamics trajectory. Now, in machine learning, employed in biophysics, you don't get the rules and inputs, but instead you feed in the machine with inputs, but as well as the output. So you actually give to the machine a data set which has some outputs of what actually happened to the system after simulating for some time. So you feed it in the inputs as well as the outputs, and then this machine is using statistical models to create the rules and to make the predictions. And I will explain a little bit more as we go on. So in machine learning, we can divide it into categories: unsupervised learning and supervised learning. Some have a third one in the classification, reinforcement learning. But well, these classifications are just schematic, and I'm sure there's many more different categorizations that can take place. So this is just schematic that I'm showing you, just to make the background. So in supervised learning, the computer is trained, as I mentioned, on data of inputs and outputs, with a goal of learning a general rule that maps the given inputs to the given outputs. So basically, you feed it in with your input and output. As we will discuss, for example, later, in my case, I'm working on predictive protein membrane interfaces. So I'm giving to the machine which amino acids are penetrating the membrane and which amino acids are not penetrating the membrane. And I expect it to learn a rule, a general rule of an unknown protein. What is the rule to predict which amino acids will then be penetrating and which amino acids will not be penetrating the membrane based on the data inputs and outputs that I have given. And we also have the unsupervised learning, where the computer is not given inputs and outputs, it's only given inputs, and then it works to discover the pattern or a structure in the input on its own. So just inputs, no outputs. And in unsupervised learning, we have usually clustering, and in supervised learning, we can split it in classification and regression. So in essentially in classification, so supervised learning algorithms are trained in data where the desired output is known, where we know the output and we want to train the algorithm to make rules. And again, this is split in regression and classification. So regression algorithms predict a numerical value based on previously observed data. I mean, I'm sure you all are familiar with regression. And in classification, the task is to train the machine learning algorithm to optimally distinguish between two classes. In my case, it's the membrane-penetrating amino acids versus the non-penetrating amino acids. So just to continue, let's see an example of supervised machine learning with classification from the field of drug discovery. So here we have a data set of active molecules versus non-active molecules for a given receptor. We build a pharmacophore model, so we know which molecules are active and for which pharmacophore model, and which molecules are non-active. And we create labels for which pharmacophores will be active. Then we input to the algorithm the labeled data set that these molecules, these pharmacophores are active, and these pharmacophores are not active. The algorithm is then being trained, and as a result, it learns rules of how to split the two classes in active compounds versus non-active compounds. And then in the result, what is given is probability of the compound being active versus non-active. And this is an example now of unsupervised machine learning, where we have, as I mentioned, only inputs. So we have descriptors that are provided to a neural network as input. So we have, in this particular case, we have bond lengths, atomic charges, and atomic positions. This is the input in a neural network. Then hidden layers compute a weighted sum of these input values, taking into account all parameters. And finally, from these hidden layers, an activation function is calculated that provides the output result, which is in this case, the energy and a homolumen. So as I mentioned, we are now at a position where we can use artificial intelligence, and machine learning is a subset of it, and deep learning is even deep subset of machine learning. We are in a position to use machine learning for our problems. And that brings me into the problem that we want to study, which is the protein membrane interface. And why we are interested in this protein membrane interface, the reason is because 50% of pharmacological targets are membrane proteins. And our lab has worked a lot in the areas of feed identification and lead optimization, on peripheral membrane proteins, as well as transmembrane proteins, always with an eye on drug discovery. So we want now to understand whether the protein membrane interface is a novel drug design strategy. And because this abnormal protein membrane attachment is involved in deregulated pathways and in disease, we think that this protein membrane attachment can be inhibited or activated depending on what you want to do with a ligand. So targeting this interface, unfortunately, has been neglected so far, possibly due to the complexity of this interface, also because the protein membrane interface is usually not known. And computational workflows to study the protein membrane interface are not tailored for this interface. And also, we don't exactly know what are the possibilities for drug discovery. Do cavities exist at the protein membrane interface that we can target? So we want to create a computational workflow for targeting protein membrane interfaces as a new drug design strategy. And we know that the approach is feasible, as a couple of papers in the literature have already proven this. So the workflow of targeting this protein membrane interface is as follows. We create protein conformational ensemble from molecular dynamics simulations. Then, as I will show you, we use machine learning to identify these membrane binding domains. And there, on this interface, we identify binding pockets. The identification of the binding pockets is not alone is not enough. So we need to understand whether these binding pockets contribute to the whole function of the protein. So we try to understand whether there's an implication of the binding pockets that we discover in allosteric networks. And once we discover a druggable site on protein membrane interfaces, then we proceed with our usual computer-aided drug design strategy, which consists on docking and scoring, so virtual screening, compound selection, and in vivo and in vitro assays. So before we start by exploring the protein membrane interface, we first need to understand the membranes on their own and the proteins on their own. And so a number of works in the previous 10 years from my lab worked on that, understanding lipid organization and cellular model membranes using molecular dynamics simulations, and have quite a few publications on that. And the reason for doing all this work, which is essentially basic science, is to understand whether the computational techniques that we have at hand are able to produce realistic results or close to experimental results, and also to make prospective predictions. And here I'm showing you one example from calculating that one lipid, which is called DPPC, and cholesterol phase diagram. And here, this is in the lines, we have the experimental result from Vistan Davis, which shows how the different concentrations of cholesterol versus changing the temperature is changing the different phases of the lipid, going from the lipid ordered phase to the gel phase and to the liquid phase, and mixtures of that. And we see that indeed, the models that we currently have, so this is the coarse grain model Martini, is able to predict very good experimental results. And here it's just a movie showing a Martini simulation of when you mix randomly in random positions lipids, cholesterol, and water, then the lipid bilayer is forming spontaneously in a few nanoseconds. But also, and we know it now that the force fields, the potentials that we use for proteins and ligands, they are also quite accurate. So here I'm showing a movie from a well-known movie from D. Shaw company, showing the cancer drug dasatinib binding on SAR kinase. So it's an unbiased simulation where the molecule is finding its crystal structure. And in studies that we also did with the transmembrane receptor 81R, we are following how the candesartan drug is binding on the 81 receptor. So it is possible to actually simulate both the lipid bilayer, both the protein ligand binding, but also the combination of all these entities together. And for us, it was of particular interest because we started around 12 years ago with studying protein kinase, lipid kinase PI3K, which is the most mutated kinase in human cancers. And this kinase is actually lipid kinase. So to produce its signal, it goes to the membrane, it's recruited by receptor tyrosine kinases, and it phosphorylates one lipid, PIP2. It transforms it into PIP3, and then PIP3 is a signaling molecule, signaling for proliferation, metabolism, and cell survival. So 80% of all the mutations of this protein happen in two hotspot amino acids, and these two hotspot amino acids are responsible for 30% of breast cancer patients. So you can understand that this is a really significant case for precision medicine. And most importantly, it's because one single amino acid change, for example, here of a glutamic acid becoming a lysine at position 545, makes the wild type an oncogene. And also histidine at position 1047 becoming an arginine, again makes this protein an oncogene. So we wondered, what is the mechanism of overactivation of this protein, and can we design mutant and isoform-specific therapies using a combination of biased and unbiased molecular dynamic simulations, virtual screening, property prediction, and filtering of the compounds, in vitro and in vivo assays, and lead optimization. And that was a study that happened, as I mentioned, about 10 years ago. And this is a proposed mechanism that we came up for the mutation of 1047R of PI3 kinase alpha. This mutant changes the way with which PI3 kinase interacts with a membrane as it accumulates more charge on the surface. It also changes the autoinhibitory role of the C-terminus, PI3K alpha wild type, into a non-inhibitory role that the oncogene creates, as well as other structural changes that are related to the mutant becoming overactive and producing more PIP3, essentially telling the signal to proliferate. And that's how it's implicated in cancer. And these calculations were also complemented with surface plasmon resonance experiments from patient-derived liposomes. Patient-derived liposomes. We also studied the other mutant, the glutamic acid becoming a lysine at position 545, and that was a collaboration with Francesco Gervasio group. And we could find that indeed, through enhanced sampling molecular dynamic simulations, we could observe the full free energy landscape of PI3K's mutant, and we could see that the mutant is actually causing more deeper minima at the open conformation, which is active. So it causes a detachment of the regulatory domain NSH2 and other structural changes that result of the activation of this kinase. So we use this information in order to proceed to precision medicine strategies and see if we can derive new molecules that are mutant specific, and they would target only the mutants and not the wild type, so leaving the patient without side effects. So in this drug design strategy, we took the results from molecular dynamic simulations, and we did the cavity recognition, binding site prediction. We saw whether these cavities are implicated in large-scale allosteric motions. And then on the cavities that were qualified as allosteric, then we did a virtual screening exercise with Glide, post-filtering with our tool, the ChemBioserver, which you can find here, and then assaying and purchasing and assaying in vitro and in vivo some compounds. This result was successful, and this work was successful. And what we did was to discover novel allosteric PI3K modulators. So here I'm showing in this first biochemical assays from the Christoforidis lab that the molecule that we use, this is an example, so PI3K10, in a low ATP concentration and high ATP concentration, the IC50 remains unchanged. So it's a very small change, which means that it implies that this molecule is non-competitive. We also show selectivity in the mutant cell lines with respect to the wild type. So this is wild type and all these are mutant. So there was selectivity. So the cancer cells were killed selectively with respect to the wild type. The pharmacokinetics allow us to have hopes because after 4 hours, there is sufficient concentration of this molecule, PI3K10, on the blood circulation of the mouse. And the most impressive result, I think, was the in vivo result, where we can show that when you treat tumors where PI3K is not involved, so tumors, breast cancer tumors that where PI3K is wild type, so it is not driven by wild type, by PI3K, then treatment with our molecule BRF41 is not doing anything to inhibit the tumor. So this is the vehicle, and this is our molecule. So you see no statistically different reduction of the tumor volume. However, when you treat PI3K driven tumors, breast cancer tumors, with the molecule, you see that compared to the vehicle, which is here, there is a dramatic reduction of the tumor volume. And this is only when PI3K is mutated. So we could prove the selectivity of these molecules. And the same is for this other mutation, the H1047R, as well. This is all xenografts here. But we also did in the Pseudotiades lab and Kinase labs a breast cancer model, which is in vivo. It's a genetically modified model, and we see the reduction of the tumors in vivo. But perhaps the most interesting result from this study was the mechanism of action of these molecules. So by doing surface plasmon resonance experiments in collaboration with Bogo Saganyan in University of Thrace, we coupled the liposomes, patient-derived liposomes on the SPR chip, and then we monitored the association of the protein PI3K to the liposome in the presence and in the absence of our molecules, as well as of known inhibitors. And the result was that in the absence of inhibitors, we have the normal high attachment of the mutant to the membrane. In the presence of inhibitors that are known, that are competitive inhibitors, and this is worthmannin, a known competitive inhibitor, then the protein association to the membrane doesn't change. So for the wild type and for the mutant, either you have worthmannin or you don't have worthmannin, because it shows the difference between the presence and the absence of the inhibitors. You don't really see any difference. But if you insert any of the molecules that we designed, you see huge dramatic changes of the way that the protein is interacting with the membrane. So this is a potential allosteric mechanism, so that in the presence of the inhibitors, PI3K is stabilized on the membrane. And this concept of allosteric modulation, even if it's at the protein interface, membrane interface, or if it's at a different site, it doesn't matter. It has several advantages because you can selectively target and inhibit mutant proteins with selectivity over the wild type. And it's non-competitive with endogenous ligands. And you can exploit all targets with new chemical space, target proteins that have been considered undruggable in the past. The most pronounced example is the first drug for KRAS, for the mutation G12C, which is targeting an allosteric pocket of KRAS. So you can target proteins that have been considered undruggable as KRAS using the allosteric principle, and also use it in combination therapies with orthosteric drugs, which may limit the resistance profile. And there was a study in Nature 2017 that showed for BCR-ABL that if you couple the inhibition of BCR-ABL with both enilotinib at the active site as well as ABL001 at the allosteric site, then you diminish the resistance profile of the tumor. You don't get resistance mutations on the protein. So these combination therapies could also be very important. Also in another study, we could find this is a GPCR, again, the AT1 receptor that briefly discussed before. We did molecular dynamic simulations of this GPCR in the presence of cholesterol. And we found that actually cholesterol spontaneously travels onto this GPCR, this AT1R, and it binds in a cholesterol consensus motif that was not discovered before. And it has this binding is actually inducing conformational flexibility to the receptor so that it can bind the drug molecules. So inside the membrane, we also have possibilities for protein modulation. We also applied this in another protein, KRAS. I already mentioned it. It's a very famous example in cancer because it's associated with 30% of human tumors. And the theory currently is that the main mechanism of KRAS is that it recruits this RAF kinase to the membrane, and KRAS acts essentially as a scaffold of a part of the RAF kinase. And when this happens, then RAF dimerizes, so it comes into dimers, and this dimerization then leads to signaling of RAF to the cell to proliferate and causes aberrant signaling. So this is a collaboration with Novartis. So we wanted to understand, because as I said, this is a theory, it's not really known exactly where is the protein membrane interface and exactly where are all these domains placed in the context of the lipid bilayer. Where is KRAS? Where is this RAS binding domain, this CRD domain? It is really not known. So we wanted to do molecular dynamic simulations to understand it. And before we did that, because this is a new project, of course, we turned to AlphaFold, the AlphaFold multimer module, but in fact, it was not able to predict the structure of the dimer. And that's one of the limitations that we found. While it was able to predict with high accuracy the structures of the monomers, it was not able to predict the structure of the dimers. It led to a very high PLDD T-score for the dimer. So we couldn't use AlphaFold. So then we started structural studies. For example, we took two available PDB codes, the only available PDB codes from the literature, basically that have this interface. Well, there are more, but these are the more dominant conformations. And we did a simulation of these two conformations. And we found in different also concentration of the membrane, because we wanted to see how the membrane will affect the dimerization. We did find that when you have KRAS on the membrane, then these PIP2 lipids may stabilize the conformation of the KRAS dimer. But when the actual part of the RAF kinase comes, which is called the effector, onto KRAS, then these effectors then stabilize already without the need of an enhanced PIP2 concentration. And we also, of course, studied the protein membrane interface, because we were wondering what kind of opportunities do we have to discover new binding pockets at the protein membrane interface. And one limitation of these studies is that these molecular dynamic simulations are in equilibrium. So we couldn't really observe big structural changes. We started from these two structures that I mentioned, but which are the most dominant. But we know that there are more PDB structures in the literature, and nobody really knows what is the actual conformation of the KRAS dimer on the membrane. So we did adaptive sampling simulations with ASMD. But we simulated for 10,000 V100 GPU hours, and the simulations really didn't converge. I mean, they went really very close to the initial model. So we think that if you want to use adaptive sampling technique with such a huge system, with proteins, with effectors, with a membrane, with water, then you need a huge computational effort. And for an academic lab, it wasn't really a choice. So we had to abandon this scheme. So we then turned to coarse graining, to coarse grain models, simply because this had worked for us in the past. So we used the Martini force field successfully for lipids and lipid self-assembly, and we wanted to use it again. Of course, this was not enough, because just using coarse graining will speed up your calculations, but it will not really give you the full free energy landscape. So we coupled it also with enhanced sampling simulations, metadynamics, which basically provide energy to the system in order to fill energy wells and explore more conformations. And again, we benchmarked extensively, because this is a new strategy and we didn't really know if it would work. We took 5 dimers from the literature: insulin, Barnase bar star, RAS-RAF, the transmembrane domain of the EGFR, and the rhodopsin dimer. And we simulated it with Martini 2.2b and Martini 3. And okay, this is a movie. Indeed, we could see that using this parallel tempering metadynamics and the well-tempered ensemble together with coarse grain models, it could really bring us the full energy landscape of the dimerization. And we could observe the spontaneous assembly of many different interfaces. Here I'm showing the dimer of rhodopsin and how it proceeds in time. And we did it for the 5 complexes. And we saw that in all the cases with using the Martini 3, we could retrieve the available crystal structure. So in hand sampling simulations with a refined Martini 3 beta force field and appropriate collective variables, we could predict the dimer crystal structures. However, these were not always ranked as the lowest energy, the global energy minimum. So while it could find it, it would not always rank it. So in an unknown problem, it would not help you in the sense that you have one structure and you nail it and that's it. It basically tells you, okay, you have 10 minima, one of them is the correct one. Now you can do more detailed calculations to find which one. But still, it limits your conformational space, especially from endless different combinations to just 10 or so minima, which you can then extrapolate. So this is still ongoing. Now we did it for KRAS, and we have promising results that I don't have time to talk about because we concluded in the end that, okay, we spent now 10 years, 12 years in these methodologies, and we had some promising results. But what if we want to actually follow the strategy that I showed you before in a high throughput way? So basically from protein conformational ensemble from molecular dynamic simulations to be able to predict a protein membrane interface. Do we really have to do all these so difficult calculations, burning thousands, tens of thousands of GPU hours? No, we decided that we cannot keep on doing such expensive calculations, since we can now exploit artificial intelligence techniques and use machine learning to identify membrane binding domains. So in our workflow, the first step is to identify the protein membrane binding domain, then identify cavities, and then identify whether these cavities are connected allosterically to the active site. So this is, I'm going to speak in my remaining time, which I see I've already spoken. So I will spend another 10 minutes to describe this machine learning strategy that we used for identifying membrane binding domains. The strategy, again, that we used is to predict membrane binding amino acids. And this is the hypothesis that we have. Our data set is on peripheral membrane proteins, the 3-dimensional structures with experimentally known membrane penetrating amino acids. After getting the input data, we extract physicochemical and biochemical features. And we use supervised learning. We train machine learning algorithms based on the features of the membrane penetrating amino acids and evaluate a predictive model based on an independent test set. So this is to predict this. So for the dataset generation, as mentioned, we took 3D structures of 54 peripheral membrane proteins with experimentally known membrane penetrating amino acids. We prepared the dataset, deleted unwanted side chains and all the different things that you do, extracted the features, and then proceeded with machine learning. So our dataset consists of these 54 peripheral membrane proteins. We split the dataset in training and validation sets and continue with data selection. We remove amino acids with the residue depth over 2.5 because this would be transmembrane. Additionally, only hydrophobic amino acids insert to the membrane, so we removed also non-hydrophobic. And then we used a test set of 11 peripheral membrane proteins. And of course, we ensure that to avoid overfitting, we have an identity of less than 40%. So the feature importance, because now we have to find which features are important for a problem. It was calculated with two different ensemble tree-based methods, the extreme randomized tree classifiers and the light GPM. Okay, I can't go into details, but if you want more information about how this works, we can discuss it later. So it was interesting that from these ensemble tree-based algorithms, we found that the important features were hydrophobicity, solvent accessibility, and secondary structure that we knew, but also we found some other physicochemical features as important that we didn't think about before. So evolutionary conservation score, flexibility, and dihedral angles. The problem was a little bit more complex because, as you can imagine, we have only 54 proteins in the data set, and that's considered pretty low. And our training set of penetrating amino acids was quite imbalanced compared to the non-penetrating amino acids. So we had to fix this by creating different balanced data sets using different algorithms. For example, oversampling the minority class with SMOT, undersampling the majority class with cluster centroids, and many different other methods. Yeah, I'm showing here all the different methodologies that we used to balance the data set. And after balancing the data set, you have to choose a classifier, and you have to choose many different algorithms because every classifier works in a different manner, and it has the potential to predict correct samples, others cannot. So you don't know a priori which is the best one. So we wanted to find which is the best, and I will show how we did this. And then you have hyperparameters, because all these algorithms have parameters that need to be optimized for the specific problem. And depending on the parameters of the model, you may underfit the data, overfit the data, or be good and robust. So this hyperparameter tuning trains thousands of models with different parameter combinations to find this good and robust classification scheme. In our splitting the data set, the initial training set was split in 5 stratified randomized folds in order to later perform this hyperparameter optimization using 5-fold cross-validation. And class balancing was done with using 6 different techniques, as I mentioned. And then in the end, basically what we did was to have 6 different training sets split in 5 different balance folds to do the cross-validation. So then we come to the supervised learning scheme. And as I mentioned, numerous machine learning classifiers exist and you want to try them. So we tried 21 machine learning classifiers using this 5-fold validation scheme. So here they are. So basically we we trained it for hundreds of thousands of times using hyperparameter optimization in order to find what is the best combination from all these 6 training sets and 21 classifiers and hyperparameter optimization, and to keep the best model for each classifier that has the best F1 score, and I will explain a bit what it is. So to decide which was the best combination, we use this meta classifier scheme. That is a voting classifier that decides which combinations give you the best sets. So I will skip maybe the technical details, just to say that the accuracy of these schemes are mentioned with this confusion matrix where you have your true positives, false negatives, and true negatives, and you combine them in different accuracy metrics that are called the recall, precision, and F1 score, as well as the MCC value, a Matthews correlation coefficient. And essentially, in our scheme, the voting classifier decides which one will be the first level classifier that is the best. And the stacking classifier trains a classifier based on the output of these first level classifiers. This takes all the combinations and then reads out a conclusion, which in our case was an F1 score of 0.86 and MCC of 0.71, which I at first didn't really look very good. But looking at the prediction, we saw that many of the false negatives that you see here are actually at the protein membrane interface. It's just that they were not labeled correctly. So we could actually change the scheme in order to classify correctly the false negatives and improve the F1 score to 0.92 and then the MCC to 0.84. And we could show that this algorithm was performing best and compared with PPM and MODA, which are two other tools for the protein membrane interface. So our model correctly predicts the membrane penetrating amino acids. This is the membrane plane here, compared to PPM, which finds the wrong plane, and MODA, which has false positive predictions in a different place. The test set prediction also showed a precision of 91% of this independent 11 proteins that we chose. And this is just another case. I can skip it. The further manual inspection of the false positive also showed that in many cases, these false positive predictions were actually not false positives, but they were at the interface of the protein-protein interactions in this particular case. So this protein is two subunits, but then it dimerizes to produce a tetramer. And this false positive was right at the point where the dimer comes in and binds. So these protein membrane interactions were concluded are most likely similar to protein-protein interactions. If you want to use this technique with AlphaFold, it's best to only take into account regions of the protein that are robust results from AlphaFold, because if you just take all the spaghetti that AlphaFold many times give you, then you have many false positive results. But if you only confine to the areas where the PLDT score is good, then you get the correct prediction. And actually, our algorithm works even for transmembrane proteins, even though it was not trained for transmembrane proteins, which was good. And it was prospectively used. We published it. I don't know if the publication is here. Yes, we published the tool, and we actually created a web server. The source code is available. And people used it. In 2022, the Sabatini group produced a paper using our predictions. From their whole complex, they predicted, and this is a publication from the actual, the figure from the publication, it's not ours. So they predicted amino acids that are membrane penetrating, and then they did the experiments to predict actually that these predictions were correct. And this was the putative membrane plane that they considered. And this is another study from the Wu lab in viruses, where again they used DREAM to predict the protein membrane amino acids, and this helped them to do mutational studies just of these amino acids and to verify the hypothesis of where the protein binds on the membrane. This was all placed in a web server, which is called DREAM. You can access it here, of course, for free and without login. This predicts the protein membrane interface. It also predicts binding sites in protein conformational ensemble, and it's implemented as a workflow that incorporates also drug discovery. And I just want to, because the time is finishing, I just want to say that DREAM also performs binding site identification using P2Rank. And you can get nice images. If you go onto DREAM, you input your structure, PDB code, or your ensemble of conformations, then you receive publication-ready visualizations of the binding pockets that are at the protein membrane interface. So there's more technical details from our study about the binding properties that can be used for drug discovery that you can all read in a next paper that we published then in bioinformatics about DREAM and how it performs using all this information and the artificial intelligence scheme, how it then reaches to the pocket detection that can be used for drug discovery. And finally, we are progressing this project even further with the master student Dimitra Paranou, who is now using natural language processing, so reading proteins as they are text. So she's using popular language models, protein language models, for example, T5, to predict these protein membrane amino acid. And the reason why we're doing it is not that DREAM is not accurate, DREAM is accurate, but this protein language model, because the pre-trained model, it really produces big speed ups in the calculations. So this is a new technology that we are using with our data set, and also using Natalie Reuters and her group's data set of peppermint. This actually, we have implemented it already, and it does predict quite good results. So in true positive is the green. Again, the false negatives is the yellow, which is actually a true positive, because you can already see that it is at the protein membrane interface. It's just not labeled correctly. But the limitation is that it's limited only on the training set. So there is a risk that you're overfitting. And in this case, it happened that the training set only has the specific protein families. And if you train your data set in the specific protein families, when you insert an unknown protein, there was no prediction of any amino acid. So with this statement, basically, I want to close this presentation, because you need to also exercise caution when you are using these artificial intelligence models, because the data set needs to be unbiased. Our in-house software, we also have more software. I'm giving this slide here in case you want to take a screenshot and use our open access and open source codes in different areas of the drug discovery process. And as a concluding remark, I would like to say that artificial intelligence and machine learning needs big data. Usually at this point, chemistry and biophysics, we still have small data compared to what you can achieve when you have billions of data set, but we're working on it. I think the community knows about it, and we're working on producing bigger data sets. The most important thing is that a data set needs to be of high quality, correctly annotated. You saw what happened with the false positive and false negative results. So when you don't label your data set correctly, your results may be mixed. And also it needs to be unbiased. You saw what happened when we had the case where the dataset was biased onto 10 or so protein families. It was limited on these 10 protein families that couldn't do any further predictions. And of course, garbage in, garbage out. If your dataset is not correct, you will produce garbage. Caution needs to be taken to avoid any overfitting. In our experience, artificial intelligence usually cannot predict a property if it is not in the training set, so don't expect it to do miracles. If you don't train your algorithm on the specific properties, you cannot retrieve completely unknown proteins. Also, AI does not usually provide physical insights, at least not yet, maybe in the future. So molecular simulations and physics-based methods are still of value because we provide physical insights that can then be fed back to this AI cycle. And I would like to close by inviting you all to the European Conference on Computational and Theoretical Chemistry in 2023. It will happen in Thessaloniki, Greece. And this is the website where the registration is open. And thanking finally the members of our group that did all this wonderful work, our collaborators, and the funding sources. So thanks again for the opportunity to become a part of this very popular mixed-gen SECAM series.
Thank you very much, Zoe. I think now is time for questions, and there are some of them. So we can do either ways. I ask the question for you, or you can switch your camera and your mic on, and you can ask directly Zoe question if you wish. So I ask Valentin to switch mic on and also camera if you want, and ask a question to Zoe.
Yeah, please.
Hello. Is it working? Thank you, Zoe, for the presentation. That's great for me. At least I wanted to ask about QMM simulations, and if they can provide some better insight of the physical properties for AI, for a training set. for example?
Of course, I mean, the highest the level of the quality of the data set that you have, the highest accuracy you will have in your predictions. So if you use a high level of quantum theory to make predictions and then feed them into a neural network or depending on your problem, then the higher chance you have that your predictions will be correct. And in the case, because I'm reading also the question, for in silico drug discovery, I guess, and whether docking is enough. Yes, of course, if you have the possibility to do QMMM studies, of course, this is a more accurate methodology, and this has to be used. Docking is popular because it takes a matter of seconds to produce the outcome.
Okay, so if can I have
yes,
a question? So basically, if you train your set like on 30 proteins or such, like by doing molecular dynamics, and could it be possible to train the AI on in silico optimized structures?
Yes,
not directly
course.
On experimental values.
But you have to remember the statement that I made in the concluding slide, that what you get in is what you get out. So if you have molecular dynamic simulations and you're confined to an energy minimum, to an equilibrium structure, you cannot make predictions for unknown big conformational changes. That's why you need the experimental work. For example, NMR can provide these conformational changes. So basically, we have to remember that the data set is crucial in what the technology can do.
Okay, thank you for the answers. That was nice.
Thank you, Valentin, because this is an open discussion within the community, how to exploit MD data for larger databases. And as Zoe said, this data should be there should be interoperability, and there should be also homogeneity among the data to utilize them for machine learning and then for suitable and robust prediction. So it's pretty interesting topic and is an active discussion within our community of molecular simulation. Thank you. Now, Andy Emerson, I'm glad to see you and ask the question, please.
Well, in actual fact, it's my colleague Bala who's here with me, and I think you.
Okay.
Hello to everyone. I think this session is also special to me because I did my last postdoc with Professor Cavalli, and Francesco did his first entry to his scientific career in the lab that I worked in Rome. So hello to both of you. My question is that, nice. If I understand from your last presentation where you applied the classification algorithms, the main problem is the data set that you have, right? You have 54, if I understood well, protein lipid assembly, and then you are using it to develop your classification problems. Since we know that in these kind of structures, artifacts like crystal packing forces, wouldn't it be more realistic to take all these 54 assemblies, do an elaborate simulation, so thereby you can have even more detailed information about the chemistry of the interfaces, and then you also by this way enlarge the data set that you have for your training. Haven't you thought in this direction?
Yeah, no, probably I didn't explain this very well because I didn't have time. So the data set is not from crystal structures. We have crystal structures, but the experimental membrane penetrating amino acids is not from structural data, is from biochemical data with mutations and so on, so that we don't have the problem of crystal packing and so on, because it's actually mutational data and protein membrane binding. The problem that we have is that you don't really, because the experimentalist tells you, oh, it's I don't know, leucine 334, but it doesn't account for the area, and then you don't have basically the neighboring amino acids, which resulted into a lot of false negatives, which were true positives, in fact. So to your first question, if we are limited by crystal packing, no, it's not the case here because it's only biochemical data. But of course, later on, if we would do what you're suggesting, which would be a great thing to do, if we would take the proteins and then do simulations, then we could actually see the whole plane of the membrane and make a new labeling of the actual, all of the amino acids that are in the protein membrane interface. So yes, in the end, if you have the computational power to go and do for 54 MD membrane amino acids, MD protein membranes, then yes.
They are from Cineca, so they can do whatever they want.
Okay. Yeah, give me your phone number afterwards.
We don't have to think so much because we have the power, so it's all right.
Yeah, exactly. But yes, in the end, if we did this and then relabel the data set, I'm sure it would reach to much higher accuracies.
Thank you.
Thank you.
Thank you.
Thank you. Camilo is, can you switch your camera and also your mic on, please?
Yeah, can you hear me?
Yes.
Yeah, thank you very much, Zoe, for this very nice presentation overview of your work, and yeah, for for showing us like these new tools that you have been developing. I was wondering about the like whether you could comment on the use of DREAM to study very flexible proteins.
Yeah.
So like IDPs or like
Yes, thank you. That's a very good question. Again, it's a matter of the training set. If you don't have IDPs in the training set, I think it's unlikely that you will get it in the end, because also these highly flexible proteins, they have different conformations. But because the training was also done in amino acid features, based on the input structure, if you give a structure that is known to be membrane penetrating, then the algorithm will actually predict it. The point here is to be able to know which is the actual, from all this ensemble, which is the structure that is membrane penetrating, and this is going to be challenging. For example, I'm thinking now alpha-synuclein, because when it's disordered, it doesn't bind to the membrane, but when it forms this alpha helix, it binds to the membrane. So depending on which structure you will give to the algorithm, it will predict the amino acids depending on what it has. So if you give it with the alpha helix, it predicts indeed that it's the alpha helix. But that's the reason why we went into this protein language models, because protein language models are structure agnostic in our case. So with a protein language model, if you have a big enough, again, data set, then you are not dependent at all on the structure. So in this case, if you have a good enough data set, and right now it doesn't seem to be the case, but if you have more data, then you could predict it independent of the structure. So thanks for this very nice question.
Okay, then is Ovakim who prefers I ask the question on his behalf. How do you validate Zoe MD simulations data?
Yeah, I think that's a great topic, and I mean I can respond, but you know, Andrea can also. It's a huge topic in our field. How do you validate the simulations? The first thing to do, and the first thing we always do in the lab, is to compare to available experimental data. And I showed you that before we went in, even thinking about the protein membrane interface, we first validated the molecular dynamics simulations on the lipid membranes. We calculated NMR deuterium parameters, for example. We calculated phase diagrams. We calculated other, for example, diffraction patterns. So the lipid electron density profiles, not the diffraction pattern, sorry, the electron density profile of lipids by layers. And then we were confident that our model is performing. The same we did for proteins and for protein ligand binding. So we did this all independently to first validate whether all the components can reproduce some basic properties, experimental properties. One of them is also the free energy binding affinity. And after that, we were able to go into the prospective application. I don't know if this answers the question, and if anybody else like Andrea, Ruben, I see here, I know also Adrian is with us. I don't know if anybody wants to make a comment.
Thank you very much. I think that validation of MD data is still an open question, an open field, and to try to exploit MD data for machine learning, we need to identify best practice for MD data validation. And you mentioned before, someone mentioned the intrinsically dissolved proteins, and certainly any machine learning engine should exploit flexible data for intrinsically dissolved proteins, because as we know, they are very flexible, they can experience very different conformations. And in this case, also what just Zoe said is not fully exploitable, because the diagram phase for IDPs is very complex and very often doesn't correspond to the one that we may have for more static proteins. So it is still an open question, is an issue that we have to address to make databases larger, robust, more robust, and suitable for machine learning-based prediction of Zoe tackled the protein membrane interfaces. Consider, for example, flexible pocket detection, which is another important field for computational drug discovery. And there is Ruben, who is an expert in pocket identification. And in all these cases, best practice in MD trajectory storage is, as a matter of fact, there is a European project running on this topic. So it's a matter of open discussion within at least the European, but I guess also the American communities. So please, Ruben, if you want to jump on this or on your question, good to see you.
Yeah, good to see you too. I was for some reason locked out for a while, so I almost gave up. And but well, great talk. And I think definitely, Zoe, it's fantastic progress. And you mentioned MODA. That's something which we developed. Originated from this ODA, which is optimal desolvation or docking area, which was initially for protein-protein interactions. And you're absolutely right that there is a lot of overlap. And then MODA was adjustment for membrane, and you showed a better performance of yours, which is of course what I would expect, because ours was based on, I don't know, 15 years old stuff. But I just still want to pitch the idea that both MODA and ODA, they're based on purely physical principles. So it just you calculate from the basic physics what is the free energy to desolvate this patch, and then it's basically it surrounds a protein with a mesh of points, and then descends different spheres of different radii, and then calculates the desolvation. So I'm sure that there. But the subtleties which you went into, I think, are fantastic because it depends on the lipid composition. It depends on whether they charge or not charge. So you will have different type of interaction, and then I think the machine learning gives you a chance to figure out what that classification of different types of interactions would be. I think that's. In terms of targeting that, it really depends. If you do interact with a charged, heads of lipids, then probably your allosteric drug would be just the something which interferes with that charge. Like stick the same charge there and it will not bind there. And you can always have, if you can get into that pocket and then get the outside part being charged the same way as your membrane, it will no longer stick there. Something of that nature, and this is a great strategy. I don't think it was exploited enough, just because of you need to identify those opportunities on the surface, and they may be flexible. Right, but you know, I think it's a great progress, and it's just the right direction.
Thank you, Ruben.
Thanks, Ruben. We should work on that more. I think more people should come into this field, actually.
Absolutely.
Right. You know, and well, you know, previously, well, because of the transient nature, well, because the nature of these interactions, and that's another part of the story, which maybe you can get into. I'm sure you working on that. Is that it's controlled, it's regulated, and some of that is regulated through phosphorylation and all the kinases, and actually in many cases, and so making that a real variable there, rather than like you know, any serine is going to interact like that, whether from like, you know, phosphorylated or tyrosine or not phosphorylated tyrosine. So like making those distinctions is probably one of the biggest missing points in all these predictors, because that's what ultimately decides whether, you know, what's going to happen, and the dynamic nature of that, and dependence on PTMs, not only phosphorylation but other PTMs as well. I think it's a missing, big missing point.
I absolutely agree.
We move to the next question from Angelos.
Yes, Dr. Cunha, I am familiar with your work in audit, and I'm greeting you and thanking you for your presentation. I have submitted a question about how you trained the DREAM model on the tetramer proteins, and how could possibly use this model in particular to study more complex structures like the VEGFR receptor. For my case, it is of interest to me.
And in which context? I don't understand.
Yeah, so I was wondering, when training data set to run the simulation, maybe should one be based mostly on the structure of the units of its protein, or of structural proteins that have a sequence which is of higher similarity to the one we're studying?
So it really, in all the different questions that we want to answer, is basically you have to define better what you want to find. What is the target here? What property do you want to calculate?
So I'm just asking for binding sites in the receptor and the membrane interface.
Yes. Well, if it's a tetramer, whether it would make a difference.
Mm-hmm. Yes. And how would the predictability of the model perform?
Yeah. So basically, if it's a tetramer and you only provide the monomer, then you will find, because this is based on structural features, you will still find the protein membrane interface of the monomer, but you will get a lot of false positives where the protein interactions are.
I see.
But of course, again, if you want to focus on particular problems, again, the question goes back to the data set and being refined for that.
I see. Thank you.
Thank you, Angelos. Maurizio, nice to see you.
Oh, hi, Andrea. Nice to see you and all the speakers. No, well, my was a kind of a provocative question, not even a provocative question, but a comment. You presented us a wonderful comparison between the classical biosimulations and the machine learning AI-based model. So, can I? Do you?
I hear you.
Oh, okay. So just my questions, and if you want to make a comment, was are we on the way to abandon the classical physics-based biosimulation in favor of just plain AI ML-based models in order to answer to the questions that we?
So
in my opinion, this is happening for organic synthesis, for example. But I don't know if what do you think about this?
I have two comments on that. First of all, I closed my presentation with your question, which I think is very important. That's why I share my screen again. So in our experience, artificial intelligence methods do not usually provide physical insights. Not yet. Maybe they will in the future. We don't know this yet. At the moment, they don't. So molecular simulations and physics-based methods are still of value because they provide physical insights. And for me, this is indispensable knowledge, and knowledge that cannot be replaced by any machine learning scheme at the moment. But this may change in the future. So I'm not sure how it will be handled. The second comment that I want to make is that I'm seeing more and more the young scientists becoming less oriented to learn physics-based methods. They want to only learn about data science and using these methodologies and exploiting the wealth of data that we have. And I see a slight disinterest of my new students in these physics-based methods, and also because of this abundance in literature, they always give me, or not always, but some of them give me the answer, why should I learn quantum chemistry? If I need to see what it is, I will look it up on Google, or I will ask ChatGPT. So I think your question is very valid, because we're at a turning point, and I don't know if it is something that we don't understand yet, how it is going to evolve, and how, if all science will disappear and be replaced by data, or it's really that we will see a collapse and then going completely back to science. I don't know. I would like to discuss with all of you.
Yeah, it's a nice thing. Thank you very much. Okay.
My motto in this respect is out-of-the-box thinking, which makes the difference between human intelligence and artificial intelligence, which is not extrapolative yet. So out-of-the-box thinking is out of any possibility of machine learning now. So stimulate younger researchers to come with very, very strange, crazy, and brilliant ideas, because this is the way to make new science nowadays. Otherwise, we cannot compete with ChatGPT, as you said. There is no competition. Probably now, yes. In 2-3 years, probably the human brain will be more limited without fantasy. Okay, Adrian, our Star Wars protagonist, please. I was not one of the authors of this paper. It's super interesting. I just click on the link you put in the chat. Your mic is off, Adrian.
Yeah, but anyway, I mean, Adrian brings up an important point. So best practices, I think we really do that. We really need it. Yeah, it was on Twitter yesterday. I also read it. Adrian.
Adrian,
you're muted. You're muted.
You are muted.
There we go. Yeah, now, yeah?
Yes.
So, yeah, first, Zoe, great talk. That was fantastic. So, yeah, that paper that I put in there was a consortium put together by Mike Gilson, and they go through, how do you validate MD data against chemical shifts and room temperature X-ray data, regular X-ray data, cryo-EM, right? And it says, and what's neat about it is that it points to public resources, right? Like validated data sets that people should be, it will be a time where we all should try to validate against the same set of data sets, right? That's going to move us forward, I think. I wanted to make a couple of comments that I've been hearing about that people have made. One of them is, one thing that I like what Zoe was doing, if you pay attention, is that I think at this point that there are many facets of machine learning AI. By the way, I hate the term AI. I hate it with all my guts. I like
the rise
machine
of
learning.
The machines. You
totally
the
hate.
rise of the machines. The rise of the machines.
No, I just don't like I don't like AI because why? First, why do we think about the I? Why should they need to be intelligent? And why do they need to be artificial? They're their own thing. So I like machine learning. That's fine. That's okay with me, right? Is human intelligence something that we want to mimic? Are we that good? Right?
So you always want to teach the machine. You want the machine to learn, but you don't want it to be intelligent. I see where you're getting at.
Why? I've seen humans. I don't like them to be intelligent. I don't like human intelligence. Let's not do that, right? Most humans are not that good, all right? Including me. So let's not
so intelligent.
So, but besides that, I don't think it's needed, right? I mean, the robots in the lab are not designed to be like a human in the lab, right?
Yeah.
They have different set of skills. And then a human building a car, sorry, a robot building a car is not designed to be a human building a car. And it doesn't have to be, right? So we shouldn't think about these things like humans. So, but the point is that if I look at what Zoe did, and correct me, Zoe, if I'm wrong, is you basically did the machine learning AI, big toolbox, and you use whatever you needed to use to answer the question that you had, right?
Yes.
And one of the things that is happening right now is that the toolbox is growing tremendously fast, and the software infrastructure, it's really available, right? If you want to say, I want to do, classification, and Zoe used, I don't know, 21 different classifiers. They're all, they're all a Python link away, right?
Right.
You don't have it's that simple, and that's really a big change, right? On the infrastructure, and that's something that it's certainly going to change the way we do things. I want to discuss a little bit about what Zoe said about machine learning not giving you insights. It can, but you have to be careful, but you have to design it in advance, right?
But I didn't say that. It did give us insights. For example, it gave us some features that we didn't think about. For example, the evolutionary score, the flexibility or the hydral angles, we didn't think about. It does give us insights. I said that it's not trained in something. It cannot generate like a completely new conformation for molecular dynamics.
Right.
That's what I said.
Yes.
But we did get insights.
I agree. That's right.
For example, the protein-protein interface that came up as a false negative, it was actually a positive for a protein. So it does give you insights.
Okay. There we go. So I misunderstood. Okay, perfect. Yeah. But again, it has to be in the design of the machine learning method and the features, right? So if you design, you need to figure out if the particular machine learning method you're using is designed to provide physical insights or not. Some of them are not. So you shouldn't even try.
Absolutely.
Right? Because you're going to go down a rabbit hole, you're never going to come back. But some of them are, right? And you should be using carefully. And the last one, like you said, it's that the machine learning models are I used to say this about force fields. Sometimes they're much, much better than they have any right to be, right? And that's kind of very dangerous because you get, one gets really excited and then, you get blinded and sometimes you don't notice a failure, right? So
yeah, absolutely.
We need to start, like you said, right? Besides training people in the basic science, we need to train people to, intuition, right? Like try to when you look at patterns, you start to look like, well, that doesn't okay, yeah, it came from an algorithm, but I know that's not right, right? Or I should look at it again.
Yeah.
With a different set of methods, because we all know it's not magic, but you know, these things are extrapolating from you know, some data
yes, and it's very easy to fall in the trap of overfitting and overinterpret your results.
Right.
Absolutely. Thank you very much, Adrian. Very interesting and insightful discussion. Francesco, can you switch your camera on now? Oh, yes. Again, good to see you.
Great to see you all here. So while
full of friends and wonderful colleagues. Please, Francesco, go ahead.
Thank you very much, Zoe, for your great talk. I have a curiosity. Out of the all the protein membrane interaction binding site that you identified, how many of them were could you predict that there were also a small molecule binding site?
Yeah, that's a very interesting question. It goes back to the comment of Ruben. Actually, in all cases, we could find pockets at that interface, and that's quite interesting because we really think that this is a neglected strategy, mostly because of the complexity of this interface and because maybe it was thought that it cannot be targeted. But in all cases, we found a pocket. As Ruben said, this is an underexplored area, and I think that it will become important in the future.
Okay, thank you. Sorry, I missed the question from Ruben. I was not there. And also, I have a curiosity since I'm here now, for the PI3K inhibitor that you found. You mentioned you showed the SPR results, and I'm wondering whether you identified, like, you generate a structural model of a 3D structural model of where your small molecule might bind if you try to, for instance, mutate the protein side, the amino acids that are supposed to interact with that small molecule. If you try to investigate a little bit more the interaction of this allosteric side.
So we did, of course, we do have structural models, but okay, I didn't show it because it's in a patent and I cannot show the structure, the binding pocket exactly, but we didn't do mutational actual biochemistry, mutational studies to verify. We had the collaboration with a crystallographer that could eventually see a picture, but these molecules are quite insoluble and we couldn't really, for me, she couldn't soak the crystal, but for me, it's quite interesting because in vivo we have good results. So the molecules really reach the target. So it is, I think it is a matter of experimental techniques, and our goal is to generate a crystal structure, and that would be, of course, the solid proof of the mechanism of action.
And what do you think is, well, because we are talking about this, what do you think is the mechanism of action is mostly or more related to the allosteric regulation towards the active site, or whether because the small molecule is interfering with the interaction with the membrane?
So what the SPR experiments showed, and it was there in the picture, but I didn't have time to discuss it, is actually that the protein is stabilized on the membrane. We thought that you would inhibit the protein membrane interaction, but this didn't happen. The protein was stabilized on the membrane, possibly in an inactive conformation. And we think that the mechanism is allosteric. So basically, somehow the conformation is frozen on the membrane and it cannot perform the phosphorylation reaction of PIP2 to PIP3. But that was a completely unexpected result because we thought that as I showed in the beginning, that you will inhibit the protein membrane interactions. But no, we stabilized it. So thanks for asking, because I think there's really a lot of physical insights that you get and a lot of interesting biology that we will see in the future based on these interfaces.
Thank you very much for your
Thank you, Francesco, for the discussion. Thank you, Zoe, again. Very, very interesting presentation. Thank you, everyone, for the very deep and insightful discussion. Now is time for the presentation from Younger Investigation, Maurice and Francesco. Please stay in the 15 minutes that you have, because in this way we can also leave some space for the poster session, for the virtual poster session. And so please, Maurice, the stage is yours.
Okay, let me share. Okay, do you see it in presentation mode? Take it as a...
Yes, yes.
Okay, so good afternoon, and thank you very much for the organizers for giving me the opportunity to showcase my work in this event. What I'm going to talk about today is how the choice of a certain restraining algorithm for the calculation of absolute binding free energies for protein ligand systems with non-equilibrium alchemical transformations influences the results, and I'm going to introduce the new loose tight restraint. Now, the motivation for this work is that we have already state-of-the-art techniques, the ones that are most used by the lab of de Groot and Gapsis, are very accurate and give very good results. But they use stiff restraints, usually Borish restraints, to keep the ligand in place. And therefore, because by a stiff restraint doesn't allow, I said by definition, doesn't allow the ligand to move, they need accurate binding post, possibly a crystallographic binding post, that are those that are usually used in the papers. Our goal is to make a technique that instead is robust to suboptimal binding poses, so docking poses. And thus we use pre-exploration, we do Hamiltonian replica exchange. And the technique by the state of the art is usually they use bidirectional. So they have both the up and the holo protein structure. We want to do it monodirectional because it simplifies the process. And in order to use, to be more permissive, we want more permissive restraints. More precisely, the technique that we used is the virtual double system single box. And it is a multi-step algorithm because I said we want to get the various conformations of the protein ligand system because we don't know them a priori. So first we have a Hamiltonian replica exchange, then we do the non-equilibrium chemical transformations, then we get the free energy. The difference, I said, with the standard is that we do a monodirectional process, we use enhanced sampling, and we have more permissive restraints. The first step, the Hamiltonian replica exchange, more precisely, we use a torsional tempering. So we explore the torsions of the system. More precisely, we have the two systems on two sides. On one part, we have the protein ligand system, where we explore the torsions of the ligand and the first neighboring residues. The second system is the ligand in vacuum, where we explore all the torsions of the ligand. In this way, we have a Boltzmann distribution of the conformations of the two systems without needing to know them a priori. This means that we can deal with competing binding poses and with suboptimal starting poses because we get the Boltzmann distributions of the various poses without a priori knowledge. Then we go doing the alchemical transformations. More precisely, for the ligand in vacuum, we extract 400 random snapshots from the Hamiltonian replica exchange, while for the protein ligand path, we extract 250. Then for the protein ligand path, in every of the 250 snapshots independently, we're going to annihilate the ligand. So we're going to need to switch off the van der Waals and Coulombic interactions of the ligand with the system. While in the ligand alone part, we're going to generate it in a pre-equilibrated box of water by switching on the van der Waals and the Coulombic interactions of the ligand with the water. In this way, we will obtain two distinct probability distributions of the unbound, so ligand alone, and bound, so ligand in protein transformations. In theory, we could calculate the binding free energy separately on the two distributions and then get them together and get the dissociation free energy for the binding free energy. But as you can see, we are doing a monodirectional approach, and therefore we have to use the Jasinski equation. But as you can see on the bottom, the Jasinski equation is an exponential average, where on the exponential we have the opposite of the alchemical work value. This means that small values of work have a way higher weight on the final results than anything else. So the left tail of the distribution has a way higher weight. So if we have a noisy left tail of the distribution, we are going to have a biased result. In order to reduce the noise, we do what mathematically is the convolution of these two probability distributions in order to obtain the combined probability distribution. Then on a more practical side, what we do is we sum together the unbound and bound work values in order to obtain the combined work values in every possible combination. Because by being done in completely separate MD runs, there is no right or wrong order. So in this case, we have 400 unbound transformations, 250 bound, and we obtain in the end 100,000 combined values. And this, as you can see, reduces the noise of the underlying distributions, and therefore gives us less biased results with the Jasinski equation. Then, during the alchemical transformation where we annihilate the ligand from the binding pocket, we need some kind of restraining to keep the ligand there. And we also need it during the replica exchange. Traditionally, we have two opposite approaches. One, the most common one, is to use very stiff restraints like the Boresh restraint. As I said, they give very good results when you know the binding pose because they keep the ligand exactly in the position you want it to be. But if you don't know the binding pose or if you have multiple, that becomes a bit more complex. So the other approach is usually to have a weak harmonic restraint between the center of mass of the ligand and the center of mass of the protein. Because this gives full freedom to the ligand, it simply makes it more difficult for the ligand to escape the pocket. I will show it better later, but we have seen that the harmonic restraint has problems converging with big and complex ligands. So we came up with a middle ground that takes the best of both worlds. And it's the loose tight restraint. So during the exploration, so the replica exchange path, we only have a weak harmonic restraint between the protein and the ligand. But then during the annihilation path, we freeze, so we block the position of one of the atoms in the ligand in order to have a smooth and reliable convergence even for complex ligands. More precisely, the atom we chose it to take a heavy atom that is highly connected and is inside the binding pocket. And if we have a rigid structure in the ligand, inside a rigid structure, like a benzene ring. If we want to see it with the language of art, during the exploration phase, we want to be like futurists. We want to get the full dynamics of the system, and we want to know everything and want to leave it free to how we want moves. But during the alchemical part, we want to be more traditional, and we want to freeze the ligand in a certain position in order to have a reliable convergence to the right result. We used our technique on a common benchmark of 11 ligands of the BRD4 bromo domain that was introduced the first time by Aldeghi et al. In 2016. We used the same force field as they used, so amber 99 ELDN, GAF 1.5, and tip 3P water. And there are the experimental values are known. At first, to know what the optimal transformation length is and to see how the different restraining methods give which results they give, we did some tests on the first 6 ligands. As you can see, with the harmonic restraint, you already obtain a reasonable mean unsigned error for short transformations, but then in plateaus at a bit more than 1 kilocal per mole mean unsigned error. We noticed that it is because the smaller and more rigid ligands, they converge to the right result very quickly, but then the bigger and more complex ligands stay overestimated. They don't really converge. Then we have the Boresh restraint that needs longer transformations in order to converge to the right result, but then it arrives to, again, circa 1 kilocal per mole, and all the ligands go more or less together. It doesn't have the problem with the bigger ones that stay completely outside. Then the loose tight restraint, by being a middle of both worlds, acts a bit as a middle ground. It still needs longer transformations than the harmonic restraint, but it managed to get a mean unsigned error that in this case is lower than the others. And it also converges with the more complex ligands, but it does not need to be tailored for every protein ligand couple, because the only thing that the user has to do is to choose the ligand to block the position. It still converges with the more complex ones, but it gives also the freedom to explore during the Hamiltonian replica exchange part. So we used the loose-tight restraint with transformations of 7,800 picosecond per transformation on the bound side on all 11 ligands of the bromo domain, and we obtained a mean unsigned error of 0.9 kilocalories per mole. And except for ligand 7, all the ligands are inside the 2 kilocal per mole error. Then we asked ourselves, which are the two most common error sources in alchemical absolute binding free energies? The first one is force field, and it's actually common in general, because almost all the ligands of the set have one torsion that is part of a conjugated system. But this is a well-known pitfall of especially older force fields like GAP 1.5. So we decided to reparameterize only the torsions with the ANI2 force field, that is a machine learning force field. And we recalculated the binding free energy for ligand 6 and 7, which shows the two because one is over and one is underestimated in our previous calculations. We can see that we obtained better results with both the ligands with the new force field, but we also have to notice another thing. Ligand 7 has still a result that is fairly similar to the one with the old force field, because as you can see on the bottom, the dihedral that is part of the conjugated system with the two force fields, it's qualitatively the same. The maxima are still in the same positions. But for ligand number 6, we can see that the difference between the new and the old force fields is of 3 kilocals per mole. And we can see that this is due to the fact that the old force field and new force field absolutely do not agree on the position that the conjugated dihedral should have. So this shows how, with whatever technique we're using, having the right force field is important, because otherwise you can have very big difference in the results depending on the force field. Then for alchemical transformations, a common problem is the binding pose. Usually you want a good binding pose, but as I said, we are trying to get a technique that is stable and robust for suboptimal ones. So again, we docked ligand 6 and 7 with the new force field with autodock. And you can see the new docket conformations are the silver one in the images. And as you can notice, ligand number 7 has a similar docking pose to the crystallographic one, but ligand number 6 is completely rotated compared to the crystallographic one. But in any case, in both cases, we obtained the results that are equivalent within error with the crystallographic pose. This shows that doing all the exploration in the beginning and then trying to get this middle ground between exploration and convergence gives us a method that is robust to suboptimal binding poses and converges also for big and complex ligands. In conclusion, we have a good agreement with experiments, a mean unsigned error of 0.9 kilocal per moles. Our technique shows to be robust to suboptimal binding poses, so docked poses. And we introduced a loose-tight restraint that strikes a middle ground between exploration and convergence. For the future, we would like to test our system on a more diverse set of complex protein ligand systems. We, of course, would like to investigate alternative restraining methods to see how those influence the results. And also, we would like to compare this method with collective variable-based approaches like Metadynamics or OPES. There is a preprint available, and for that, I would like to thank Piero Procacci from the University of Florence and Francesco Luigi Gervasio, my PI from the University of Geneva and UCL in London. And of course, I would like to thank all my research group, all my colleagues. Check out the preprint, and are there any questions?
Thank you very much, Maurice. It's time for question. If any, you don't need to write the entire question, just put your name on the Q&A session and I will give you the stage. I can probably start with a very, very general comment, Maurice. In your perspective, I will probably add one prospective test. Since you are working in a pharmaceutical science department, I would suggest to consider the possibility to make a drug discovery prediction with completely new ligands. And another more technical point, particularly for one or two ligands I have seen in the list, be careful about the pKa, because for the final partition function, you need the right population of each protonation states to be accounted for in the proper percentage as dictated by the pKa of those ligands, which is not trivial because the pKa calculation is another very, very open field of discussion. There are some acceptable methodologies, but still far away from a good and accurate prediction. I don't know if you want to comment. Were more comments than questions, but if you have anything to add.
For the novel ligands, the technique is already usable for novel ligands, and the code needed to do the pre and post processing and setting up the system is available on my GitHub. The link is in the preprint. We use this test system because we already, in this case, we already had the experimental values, and also because, as you said, for the hydrogens, it was already used with success in multiple techniques. So we know that the protonation states are good enough for getting a good result. Because of course, when you have very complex protonations, if you're lucky, there is one main protonation. By using those, that one, you get the right free energy. Otherwise, you will have to come up with something like doing this calculation separately and doing an average, but you need to know the proportions, or you will have to come up with something new. But in this case, we used the same protonation as in the original paper because we knew it was good enough.
Okay. I don't see any other question. Thank you again, Maurice, very much for being here and for delivering this lecture. Now, Francesco, the stage is yours. We move to a different topic, still prediction via... Adrian, would you like to ask a question?
Maurice, that was a great talk. I actually follow I read the paper, the markup. I liked it.
Thank you.
I have a, first, contact me separately. I like that you use Annie for retraining the dihedrals. Contact me to see if we can figure out those torsions. I think I know what's going on, okay? So we can try to help with that. But a question, right? So you show the convergence versus time, right? Yeah. To train, but I think you agree with me. You didn't show this plot, right? But in the preprint, the correlation between the estimated delta G and the experiment was not good, right? You got R values of 0.08 or even negative, right?
You mean on the final results or during when I was calculating?
No, when it was just the initial 5, right?
On the initial 5, they're also all similar in binding free energy values because they're in order. And so by being very close, it's like trying to fit a line through a point.
Right. But what I mean is that you show the plot of the mean and sign error versus the length of the simulation for different types of restraints. Yeah. And that's basically... Yeah, I mean, it's correct. It goes down, etc. But really, the correlation was not great, right?
For
it was not a great set of points to make a decision. Do we agree on that?
Well, like I can see, maybe there wasn't a great value in there, but then I showed that it was good enough with putting the rest of the setup. But in any case, for the loose tight restraint at the longer ones, it was good. Then of course, by being very close in binding free energy, there was no way to get like a nice to fit a nice line through it. So of course it.
Oh no! In the what I'm saying is, once you put the 11, it's actually fantastic. What I'm saying is that the decision you made with initial how many 6 or something, right? It was surprising that using those 6, then you made a decision and it ended up working okay. It's nice.
I chose to do the hard way because I put the only two charged ligands of the set. I've put them in number 6, and I put the bigger one of the set, and so I really have a diverse one. Then, of course, why I've chose 1 to 6 and not 6 to 11, it's been because they were the first 6 ones.
Yeah, no, it just happened to be that you picked the one that experimentally were very, very close, right? So
yeah, because like I wanted a diverse set, I wanted the charged ones in order to show that I took them into consideration.
It's actually, it's a very nice piece of work. I like it.
Thank you.
Congratulations. Good. Okay.
Thank you, Adrian. Thank you very much. Francesco, please.
Yep. Good afternoon, everybody. Let's share my presentation. Yeah. Okay. What? Oh, sorry. I don't know what is happening, but I cannot share it. Yeah. I don't know what's going on. Sorry.
Can you see, Francesco, the green bottom on the bottom of the window? Share the screen.
Yes, I'm trying to do that, but it seems that no way.
Okay, you make it.
Can you see? Okay, good. Sorry. Okay.
Yes.
So I think you can see my slide now. Yeah.
Yes, we can see your slide, and please go ahead. Your face is on the bottom of the presentation in a kind of
shade
roast presentation. But anyway, go ahead. Otherwise, okay.
Yeah. Okay. I think we can reduce it. Okay. Sorry again. I'm going to talk about the work on the ribosome and the simulation that we perform on the ribosome, and in particular on the stalling mechanism induced by the XBP1U translational arrest peptide. Okay, so in a nutshell, probably you all know what a ribosome is, but to recap very briefly, it's made of proteins and ribosomal RNA. It's the site of where the magic happens, where the proteins are synthesized inside the cells. And at this site, the mRNA is translated into the corresponding sequence of the amino acids using tRNAs. In this particular case, in the case we would like to study, we have that the human ribosome is arrested by means of a Nash chain So inside the exit tunnel of the ribosome, there is the Nash chain that is synthesized along the way, and due to the interaction between this Nash chain and the exit channel, the ribosome is put in a stalled state, and this has been done for regulation purposes mainly, like alternative splicing and membrane targeting. This particular Nash chain is involved in the unfolded protein response, so it's crucial for the survival of the cell. What we have done is we have taken the structure of the entire ribosome with the 24 amino acid Nash chain inside the exit tunnel. It acquires a quite peculiar conformation in the exit tunnel with a turn inside the exit tunnel very close to the peptidyl transferase center. And what we have done, at first we have simulated at the equilibrium this very big macromolecule. But then, in order to understand the mechanism of action of the arresting peptide inside the exit tunnel, and the effect on the ribosome, we have switched to non-equilibrium simulation in the presence of different arresting peptide variants. What was our strategy? Because of course, it would be impossible for a Nash chain to exit from the exit tunnel along an equilibrium molecular dynamics, even if we might be able to do it for a very long time. So our strategy to try to escape from the arrest of the ribosome was to gently pulling out the peptide by means of adiabatic bias molecular dynamics. And this is a sort of polar ratchet simulation in which practically what happens is that the bias along the simulation is null if the system try to move towards the desired ending point, whereas it dumps the fluctuation if the system attempts at moving in the wrong direction. So imagine that you want to see the Nash chain exit from the exit tunnel of the ribosome, and so if we select an ending point at the bound of the box, and we instructed the simulation, so the Nash chain in particular, to reach that point. Unfortunately, I'm not able to show you the video, but let's go ahead. Practically, we have made not only the simulation for the wild type arresting peptide, but also for 4 variants. We have chosen and modeled these variants accordingly to mutational analysis in 2 experimental papers. And in the end, we choose single, double, or also triple mutants of the arresting peptide. And okay, we set up our protocol. At first, we have done a long 1 microsecond simulation of the ribosome in the presence of the arresting peptide inside the exit tunnel, and it was very stable. And this simulation was, in the end, was 2.5 million atoms. But then we model also the 4 variants with the relative mutation. So in the end, we have done a preliminary set of adiabatic molecular dynamics simulation looking for the system-specific spring constant. And then we performed 20 replicas of adiabatic MD for all the 5 variants. So in this case, due to the fact that the whole Nash chain has to be solvated and to be completely extruded from the exit tunnel, we were forced also to increase our simulation box. And in the end, the simulation was a simulation of 3 million atoms. And we have also chosen to restrict our simulation time at 100 nanoseconds using this one of the particular the system specific spring constant to understand which were the arresting peptide that were able in this amount of time to exit from the ribosome and the ones that were not. So this is the arresting peptide inside the exit tunnel. We have done a structural analysis of the stable microsecond long simulation. We identified some intramolecular, intermolecular network interaction between the arresting peptide and the ribosome and the protein that compose the exit tunnel of the ribosome. And in this way, based on this interaction, we were able to understand what would have been the results of the following adiabatic molecular dynamics. At first, we of course, like I can show you in this plot, we have plotted the collective variable progression along the adiabatic molecular dynamics simulation. But as you can see, it's not really possible to give a quantitative analysis of this of the progression of the collective variable, except for the mutants with at position 247, in which surprisingly we have seen that due to the presence of the lysine at this position instead of a cysteine, we have seen a completely stuck simulation in which the arresting peptide was not able to exit from the exit tunnel of the ribosome. Going a bit more in details and analyzing a bit more quantitatively our simulation, we have plot in this graph the time that is needed to detach for each amino acid of the arresting peptide residue from its original position, so from inside the exit tunnel, and to move out from the ribosome. And as you can see, in the presence of some mutation at position 247 again, and at position 254, we have a very strong effect in terms of the delay that has been seen for this amino acid to detach from the ribosome exit tunnel. Again, the previous plots in which nothing was happening and the arresting peptide was stuck inside the simulation, inside the exit tunnel, are represented by this dashed line in cyan, in which we have that apart from the first amino acids, the other ones were not able to detach from their original position. Let's go ahead. And yes, this is the detailed analysis that we have done in terms of, again, inter and intermolecular interaction. We have found the reason why, for example, the simulation containing that huge effect with the lysine not able to detach from its original position, that were in which the intra and intermolecular interaction were very stable and were not detached at all, were not disrupted at all, and as well as we have understood which were the hinges of the arresting mechanism. So in terms of cause and effect between the mutation that we have done on the arresting peptide and the interaction that were disrupted or reinforced. So as you have seen, we have ranked the wild type arresting peptide and the 4 variants with the different mutation in terms of detachment kinetics. We have analyzed the crucial aspects of the complex interaction between the ribosome and the arresting peptide, and we have captured these crucial aspects. We have revealed which are the main interactions happening between the ribosome and the Nash chain, this particular Nash chain, but that underlying the arresting mechanism. And we have also detailed in terms of cause and effect, the molecular explanation between the point mutation and the arresting peptide potency, depending on the arrest and the mutation that we have introduced. So what's next about this project? And probably by means of, for example, of some well-known tools, we should be able to scan the ribosome in order to look for putative ligandable pockets. And at the same time, why not try to run a drug discovery design campaign based on this ligandable pocket detection prediction in order to, for example, specifically target non-wild type ribosome, for example, in cancer. So this is our ideas and what we have done recently. And I want to thank the collaborators of this project, of course, for supporting the discussion and for writing this wonderful piece of science that we have published. And of course, I want to thank Andrea and the members of our group at IIT for the opportunity, and of course for the second for the opportunity to present our work. Yes, thank you for your attention, and I'm waiting for your question.
Thank you very much, Francesco. Now, questions from the audience. I'm seeing the number of attendees decreasing quite importantly. I can understand because we were super late, and everyone has probably different engagements, different activities to carry out. Anyway, a usual question, just to trigger the discussion, even though I'm one of the authors of this paper, is usually why such a big system wasn't simulated using Martini or coarse grain methodology, which are typical for this kind of system, and which is the added value of atomistic simulations, of the treatment of an atomistic level, such a big molecular complex.
At the beginning, there was the idea to simulate with a different approach this big macromolecule, but the fact that we were interested in understanding the molecular interaction between the arresting peptide and the ribosome, as well as to understand all the process going on the ribosome that induced that the stalling were forcing us under a certain point of view to consider the whole ribosome and at the finer grain details, because of course otherwise it would have been easy using a coarse grain approach to lost some crucial interaction between the arresting peptide and the ribosome. And at the same time, considering the non-equilibrium simulation, introducing also the variable of a coarse grain model would have been risky in terms of possible quantitative interpretation of the data and quantitative results from the simulation.
Thank you, Francesco. I'm seeing Xavier. Nice to see you. Good afternoon, and please.
Okay. Hi, Andrea, and thank you, Francesco, for the talk. So it's really impressive. I mean, the nature of the simulations that you have presented in your talk. The question that I wanted to pose is something related to what Andrea remarked before, because you have mentioned in your talk the simulations with two different force fields, the amber 14 and char 36. And since we are talking about a nascent pentide without a well-defined structure, I would like to know whether you can comment a little bit about the similarities in the results obtained with the two force fields.
Okay. Well, initially we have done the simulation only with the amber force fields. And then, due to some request from the reviewer during the publication of the paper, there was a review that was asking us to make a comparison between with the use of the charm force field. So we have selected only two systems, two variants of the arresting peptide, the wild type one and the one that was giving let's say not the full stalling, like the very strong stalling variants, but the one giving a mix of results with some replicas giving some stalled state, instead someone else, some other replica exiting, making up to exit from the exit channel. And what we have found is that in terms of time to the touch from the exit tunnel, there was not different at all. Well, in terms of comparing the same variants between the two force fields, I mean, comparing the wild type between the two force fields and the variants, the mutants with the two force fields, what we have seen was that with the charm force field, that there was still a strong stalling between the induced by the arresting peptide, and the interaction between the arresting peptide and the wall of the ribosome were stronger than the one with the amber force field. So let's say that the force field was inducing more stalling than in the case of the charm force field on the ribosome than in the presence of the charm force field was increasing the stalling somehow. Was the interaction between the arresting peptide and the wall of the ribosome were stronger than with the amber force field.
Okay.
But the general behavior was conserved. I mean, the wild type was easier to detach from the ribosome exit tunnel, and the mutants instead was also in the charm force field case increasing the stalling strength of the arresting peptide, but in more in details, the interaction were stronger with the charm force field. Yes.
Okay. Thank you, Xavi, for raising this point, because to be quantitative, also connected to the talk of Maurice before, as people, as scientists involved in computational drug discovery and computational biophysics, we would like to get some quantitative data, free energy kinetics. And for such big systems, which are those for the next generation drug discovery, even changing only the force field, you can get different outcomes. And So we have to be very careful in providing numbers. We are among computational people. So when we talk to experimentalists, probably can say something more, but among us, we should be very aware that reporting numbers in a quantitative way is maybe a delicate point based on this kind of simulations, particularly for such large systems. Thank you again, Xavi, for being here and for asking questions. Maurizio?
Here I am. Thank you. Great job. Impressive simulation that system. Quite shortly, you mentioned in your, I think in your last slide that this system might be the target for drug discovery, for designing some kind of drugs. Could you comment on this and say something about that?
Sure. The idea is to target specifically the oncogenic ribosomes. Let's say, if the ribosome is involved in the development of a cancer, this could mean that the ribosome itself is mutated. So it would be interesting from our point of view to specifically target that ribosome, the ribosome with the mutation, let's say. In this way, it would be possible to specifically target that ribosome and leaving the wild type target ribosome alone, let's say. So in this case, it would be a way to specifically target the ribosome of the cancer and not of the rest of the body. So if the ribosome is mutated and you are able to target that particular pocket, including the mutation, it might mean that you are able to distinguish between a mutated ribosome and a non-mutated one. So if you have a cancer somewhere and your ribosome are involved, those ribosomes are involved by means of mutation in the development of the cancer, of course, you are able to block only those particular ribosomes and not the ribosome of the rest of the body.
That's interesting. Looks like a bit challenging, but, yeah. But I think you are on the right way doing that. Okay, thank you very much.
Welcome. Thanks for your question.
Thank you, Maurizio, for the question. And if there are no more questions, I think we can close here the webinars. Again, thank you very much, Zoe. Thank you, Maurice. Thank you, Francesco. Very impressive talks. Very different topics, and still in the field of computational biophysics and computational drug discovery. I think we can now just stop for a few minutes and then we can jump into the virtual poster session. Thank you very much again to all the attendees, and on behalf of SICAM, I would like to invite you for the next MixGen webinar, which will be in a couple of weeks. Thank you again. Bye-bye.
Thank you. Bye.